Welcome to This Explainer. Today, we are going to unravel a topic that touches almost everyone's daily life, yet honestly, remains pretty deeply misunderstood. We're going to demystify the science behind genetically modified organisms, yep, GMOs, and explore exactly how modern biotechnology is evaluated through an ethical and religious framework, specifically looking at Islamic dietary laws. It's this absolutely fascinating collision of cutting-edge molecular biology and ancient jurisprudence.
And really, this is the core question we're answering today. When science alters our food's DNA, how do we actually know if it's safe or halal? To figure that out, we're going to trace the journey of a modified gene from its origin in a laboratory all the way to our dinner plates. Because as biotechnology creates these completely unprecedented combinations of life, understanding the underlying science is literally the only way to accurately evaluate the ethical and dietary implications.
All right, here is our roadmap for today. We'll quickly cover the basics of biotechnology, how genetic modification actually works, look at some real-world GMO examples, see how we detect GMOs in our food, and finally, dive into the halal perspective. Let's get into it.
Starting with Section 1, the basics of biotechnology. We absolutely have to lay down the biological building blocks before we can get into the bigger applications later. You really can't understand the ethical rulings without grasping the science first.
The crucial point here is getting our terminology straight. Biotechnology is actually a super broad term. It's defined simply as using live organisms or biological systems in industrial processes. Even things like waste treatment plants fall under this. But genetic modification is much more specific. And when we use the word transgenic, that refers to plants or organisms that have been intentionally changed by adding genetic material from a completely different species.
Think of this as the biological master blueprint. It's known as the central dogma of biology. It shows how the instructions locked inside that stable double helix of DNA are transcribed into a single strand of RNA, then that RNA is translated to build proteins. Those tangled protein structures are what do the actual physical work and determine the traits of an organism. Simply put, if you change the DNA, you change the resulting protein.
Moving on to Section 2, how genetic modification works. Let's see how this blueprint metaphor translates into the actual physical process of changing an organism's genetic makeup in a lab. The precise procedure to seamlessly cut and paste a desired trait happens in 3 steps. First, genes are removed from a targeted DNA strand using specific enzymes. Think of these like incredibly precise chemical scissors that recognize exact sequences. Second, using other enzymes, this donor gene is integrated into the recipient organism's DNA. And third, we have gene expression. The newly inserted gene basically falls under the control of the new host, and it starts expressing the new protein, allowing it to perform its totally new function.
Section 3. Real-world GMO examples. Let's bring this science out of the abstract and see how these techniques are actually deployed in the field to solve massive agricultural problems.
We're going to look closely at the case of BT corn. It all starts with Bacillus thuringiensis, or just beet for short. This is a really fascinating, naturally occurring bacterium found in soil. During its spore formation, beet produces these specialized crystal-like proteins. And what scientists discovered is that when these specific proteins are eaten by certain insect larvae, they act as a highly targeted poison.
What you're seeing here are the microscopic cell structures inside the Bacillus thuringiensis bacteria. When these are harvested, they act as a potent insecticide. They specifically damage the digestive tracts of pests like caterpillars, beetles, and corn borers, but, and this is key, they leave other organisms completely unharmed. Now, for years, farmers would just spray this bacteria on their crops, but traditional sprays wash away in the rain or their toxic effect degrades under UV light and heat. To bypass that problem entirely, scientists isolated the specific BT gene responsible for the toxin. Using vectors like plasmids, they insert it directly into the embryonic cells of the corn itself. Those cells are grown in petri dishes, and the successful ones grow into full corn plants that inherently protect themselves. Talk about a game changer.
But it's not just about pest control, right? Scientists have engineered tomatoes like the flavor savor by reversing a specific sequence called the PG gene. This dramatically slows down softening and rotting. In another incredible application, scientists created golden rice by inserting genes that fortify the rice with beta-carotene, which is a precursor to vitamin A. This rice is specifically designed to be fed to children in developing nations to increase disease resistance and prevent visual impairment.
Section 4. Detecting GMOs in food.
Once these modified crops leave the lab and the farm and enter the global food supply, we need rigorous ways to identify what is actually in our food.
We basically have two main ways we test our food. The DNA-based method, using tools like PCR, targets the DNA itself. Because DNA is highly stable, this process is generally pretty simple and straightforward, perfect for detecting meat adulteration or identifying specific species. On the flip side, the protein-based method, like ELISA, tests for the proteins the DNA creates. Because proteins are dynamic and fragile, or labile as scientists say, this process is way more tedious and laborious, though it is super useful for detecting how an animal was treated before slaughter.
Which brings us to Section 5, the halal perspective. Now that we deeply understand the modification process from a molecular and agricultural standpoint, it's time to pass these scientific innovations through the filter of Islamic dietary laws.
To really grasp this intersection, we have to recognize a fundamental dichotomy here. The scientist provides the objective facts. They explain exactly how a gene is transferred, like how do you get a fish gene into a strawberry or a soil bacteria into corn. The religious scholar, on the other hand, doesn't debate the science. Instead, they take those facts and interpret if that specific development violates any of the tenets of Islam.
Islamic scholars use several guiding principles for this. One of the most powerful is this quote: "Whatever halal is sufficient and whatever haram is superfluous." Basically, haram is usually associated with what is harmful and unhealthy. Interestingly, from a regulatory standpoint, if government authorities determine beyond a doubt that a GMO is harmful, it won't be approved. And if it's harmful, it automatically fails the halal standard as well. Good intentions never make a haram thing halal. If there's doubt, you avoid it.
So how do these principles apply in practice? The rulings actually reveal a highly nuanced approach. A plant-to-plant gene transfer, whether it's for making ingredients like citric acid or changing crops where the genetic material is actually consumed, like corn and rice, doesn't really pose any issue. It's generally considered acceptable. However, transferring animal genes to bacteria to manufacture enzymes, that requires strict conditions. It's only acceptable if the safety of those ingredients is established beyond a doubt, and the entire production process adheres strictly to halal standards.
By tracking the journey of a single modified gene, from precise enzymatic cuts in a petri dish to a fortified grain of rice to the rigorous ethical debates of religious scholars, we're left to ponder an incredible question. As biotechnology creates unprecedented combinations of life, how will ancient religious tenets continue to adapt and guide our ethical choices? Science is absolutely going to keep pushing the boundaries of what's possible, but ultimately it's our ethical and cultural frameworks that will decide what ends up on our plates. Thank you so much for joining me on this explainer, and keep questioning the science around you.